Welcome everybody to the 16th edition of the Multiomics Reading Group. I'm your host for today, Cesar Miguel Valdez Cordova, and today we have the absolute honor and pleasure to present Brian Clark. For a little introduction, Brian's research centers on combining machine learning and mathematical modeling with rigorous statistical principles to understand core questions in genomics. One focus area is predicting effects of rare genetic variants for disease risk stratification and discovering gene-phenotype associations. He also develops causal inference methods to analyze the dynamics of gene regulatory networks in developmental systems, with the goal of understanding cell fate decisions and disease-relevant pathways. Prior to working at the German Cancer Research Center, he did his PhD at the Max Planck and further postdoctoral work at Stanford and the University of Münster in mathematics. Following that, he has worked in data science and machine learning in industry. In his spare time, he also enjoys spending time with his family, bouldering, rock climbing, cycling, and hiking. So without any further ado, please, Brian, take it away.
Yeah, thanks very much for the nice introduction. And thank you as well for the invitation. Before I start, I want to give a sincere apology. Actually, I just noticed on my slides, I don't know if you can see my cursor, but I just noticed that I still have the original date that my presentation was supposed to be on March 12th. As I guess you know, there was a bit of a mix-up with the daylight savings time because you had switched to daylight savings time already and we hadn't in Germany, and I completely confused what time I was supposed to be presenting. So apologies for not showing then. I hope the presentation will be interesting enough to make it worth the wait. Here we go. So I'm going to tell you about this work that we did and published in Nature Genetics last year about using deep set networks for rare variant association testing. Just to give a little bit of context for what this is, we are trying to understand the link between genotype and phenotype. So, for example, a lot of what is done in genetic studies is that you have like a genetic variant at a certain position in the genome, and you want to understand how that affects some phenotype. A very simple example would be someone's height. So, if you have there are some genetic variants where if you have them, they cause you to be a little bit taller because one bone is a little bit longer or something like that. Of course, you all know that when it comes to doing machine learning, the sort of classical way and the way that most people do this in genetics is that you use statistical learning. So you use some rigorous methods from which you can, which are, very simple in terms of what kind of functions they can express, but you can do a lot of calculations with them. For example, you can get p-values from them so that you have some estimate of how robust your association between the genotype and the phenotype is. But of course, there's this whole other world that we're here to talk about today as well, which is deep learning, where the whole point is to stack layers upon layers and get layers, which is a bit less rigorous, but much more expressive in terms of the functions that you can do and in terms of the predictive accuracy that you can achieve. And the question that we're going to have in this talk is, can you take this business world of statistical learning and the party world of neural networks and deep learning and combine them to get the best of both worlds? So that's what we're going to try to do. Just as an outline of what I'll present today, I'll give a little bit of a background on rare variant modeling since it might not be familiar to everyone. I'll go through the model that we've developed and talk about how we apply this to rare variant association testing. And then finally, at the end, I'll give a little bit of results when we apply this to the phenotype prediction task as well. And finally, I'll give a little bit of outlook for where we're going with this. Okay, so starting with the background, I'm not sure if everyone is familiar with the concept of genome-wide association testing, but I'll try to review it here. So the standard idea of GWAS is that you do something which you do essentially per variant association testing. So what that means is you have a trait and you look at a single position in the genome, and maybe most people, or at least in the reference genome, this position has in this position you have an A nucleotide. But maybe some people have a C there, and you can either have one copy of the A and one of the C, that's called heterozygous, or you can have two copies of the C, which is called homozygous alternate allele. And maybe this has an effect on whatever trait that you're looking at. So having one copy causes the trait to reduce a little bit, and having two copies causes it to reduce roughly twice as much. Most of these effects in genetics are additive, though not all. And the way that this is typically modeled is you try to model your trait here for your individual eye as, well, you control for some covariates, which we call X. These are essentially just controlled variables. We learn a weight for these things, but we're not super interested in them. Usually it's things like age and sex, and it might have something to do with their genetic ancestry and so on. But then we take this single variable, which is how many copies of this variant do you have? So here this Gij for someone with two A's would be 0. Here it would be 1, and here it would be 2. And we learn a weight beta J for that variant J, based on just by doing normal linear regression. Since it's linear regression, we get an effect size. So we essentially know, how much is the trait going to change on average, given one or two copies of the variant. And you can also compute a p-value. So the p-value tells you essentially whether you are justified in rejecting the null hypothesis, that the null hypothesis being that the variant has no effect. And the typical way that this, or sort of the classical way that this is done, is that you study relatively common variants. The definition of common depends on the size of your data set, but might be like an allele frequency of 5% or 1% or something like that. And since you study usually about 1 million of these on what's called a genotyping array chip, you have to reach a like genome-wide, what's called genome-wide significance, which essentially means that your p-value after multiple testing correction is less than 0.05, this sort of arbitrary threshold that everyone uses. You actually have to achieve a p-value once you do the typical Bonferroni correction. You have to achieve a p-value that's actually less than 5 times 10 to the negative 8. So you have to have a really, really, really clear effect in order to achieve this genome-wide significance threshold. By the way, if there are any questions, please feel free to interrupt me also in the middle of the talk. Just speak up. I cannot see the meeting screen, so you'll have to just speak up on the microphone, unfortunately. Okay, yes. And the reason that you do this, I mean, this seems a bit weird to people who are working on machine learning. Why don't you, the question might be, why don't you just throw all the variants in and try to predict someone's phenotype and then examine your model after that? Well, the reason you do this is generally that you're not so interested in doing risk prediction, although you can get to there. And we'll talk about that at the end too, but you're more interested in understanding what are the genetic underpinnings of disease. So once you've actually, so the advantage to doing it this way is that you have a direct link between the variant and whatever disease you're studying. And then you can use this to understand, what are the genetics of that disease, what genes are affected, and so on. And a big advantage is if you're doing drug trials, then having genetic support for the target of your drug gives you a pretty large increase in the chances of success for your drug trial. So on the order of about 2.6 times. It improves with increasing confidence in the causal gene, which can be a bit difficult with rare variants because many GWAS hits are non-coding. So they're in a non-coding region of the genome, and then you have this question, which is not so easy to solve, of how do I decide which gene this variant is affecting. Okay, we are not going to look at common variants so much here. Actually, what we're interested in this work is rare variants. And there are two reasons that we're interested in rare variants. So one is that coding regions, the regions that have very clear links to the gene that the variant is affecting, these are actually more highly conserved. So you don't see as many variants in those regions, or better put, you don't see, if you have a variant in that region, it's less common in your population. So it tends to be what one would call a rare variant that needs to be studied with different methods than what you would use for common variants. And also variants with large effect sizes tend to be rare. This is especially true for variants linked to disease because these things are under selection pressure. So, in the past, people who had variants that cause them to be more likely to get a disease might not be as, likely to reproduce. And you can see this here in this actual GWAS of height that I have here. So these are all variants that have been, some of them have been linked to various genes. And these are all have significant effects on someone's height. And you can see that, so as we go right along the x-axis, the variant is becoming more frequent or more common. As we go left, it's becoming more rare. And there's this sort of inverse relationship between the frequency of the variant and its effect size. So there, some of these variants can affect your height by almost 2 centimeters, just having a single mutation in one location of your genome. Yeah, it's clear why you wouldn't have a ton of these, because then people's heights would vary wildly all over the place. But it's much more, I guess, height is not as fitness related, but it's more clear when you think about disease. Okay, now the problem is that if I were to try to apply GWAS to very rare variants, I would run into a couple of issues. The first is that you have many, there are many variants that are super rare, right? So we're using data from UK Biobank, which I'll talk about later. And in this data, so we're using sequencing data, and the most common type of variant we see is a variant that's only found in one individual. Which makes doing statistics really, really hard, like if you have a sample size of 1. And the next most common type of variant we see is one that's in 2 people, and then 3. So it follows this sort of power law distribution, more or less. So you have to deal with this problem of super rare variants, which means your data is very sparse. And the other problem is that you have a much higher multiple testing burden than you do in GWAS. So if you remember when I was talking about GWAS, you might be looking at about 1 million variants. On the UK Biobank data set that I'll talk about later, we have more like 26 million variants. So you have a much larger number of variants, which leads to a much larger multiple testing problem. When you combine that with the fact that you don't see the variants very often, it makes it very hard to achieve, statistically significant p-values using rare variants in with the traditional methods. And so the solutions that people do is they do region-based testing, which means you aggregate variants together into a region, and then you test that region against the trait instead of testing for a correlation between an association between the variant and the trait. And the region is usually can often be on the level of a gene. So now instead of testing for associations between a single variant and a trait, we're testing for associations between any genetic variants in a gene and the trait. And the other thing that you do is you can use functional annotations. So these are essentially annotations of the variant effect or other aspects of the variant, such as its frequency in the population. And you can use these to try to sort of filter and weight your variants so that you can understand their effects better. And the classical way to do this is called burden testing. And what you do in burden testing is first, if you have a set of variants in the genes, so here on the rows, these are sort of different individuals, and these are the exons of the gene. In burden testing, you first filter out variants that you believe to be harmful. And then you check whether there is a difference in the prevalence of these variants between your cases and your controls.
Sorry, Brian?
Yes.
We have a quick question in the audience.
Please, yeah.
So in terms of the region-based testing, is it always community?
Oh, I'm sorry, I can't really hear. It's very quiet.
Okay.
Is the mic on? Is the mic on? Sorry, I didn't.
Okay, sorry about that.
Better. Yeah,
in terms
thank
of
you.
region-based.
Sorry, I lost you again.
Can you hear me okay?
Yes, I can hear you now. Sorry, I just
Yeah, sorry about that.
try to find the meeting window. Yeah.
Is it always linear distance along the genome, or do people do fancier things like topologically associated domains to link the variant to the gene? Or
Oh, that's a good question. I've never seen anyone do that. It's not a reason you shouldn't, but I've never seen anyone do it.
So it's always like within the megabase of a gene or something like that? Like
Yeah, so it's usually like, I mean, the most typical thing that you would see is like the coding region of the gene, maybe a little bit of overhang, like the core promoter region or something like that. But sometimes what people do is they'll just tile the genome into like, 1 megabase regions or 100 kilobase regions or something like that. Yeah, so that's a common thing. Or if you're looking for like rare variants that affect expression, then you might do what you said, like look at the gene plus or minus n base pairs around it.
Thanks.
Yeah, sure. Okay. Yeah, thanks for the question. I'm glad. It's nice nice to not just talk to myself. Anyway, okay. Yeah, so what you do here is you calculate what is called a burden, and this takes the place. I mean, if you look at this equation, it's exactly the same as the one I showed you for GWAS. It's just that instead of looking at the count of variant, the allele count of the variant in that individual, I look at this burden, which can be 0 or 1 or 2, or in principle it can be 10 or 20, but usually most people don't have that many rare variants. And you can also count these up with a weight. That's often what people do. Instead of just counting the number of variants, you can count them weighted by, for example, 1 over the allele frequency or something like that. But the point is you hand design these filtering and weighting functions. And I don't need to tell machine learning people that this is a big red flag, that humans are generally bad at designing these filtering and weighting functions, and machines are usually better. So yeah, there are other more complicated tests you can do. There's something called variance component tests, which is quite common. I encourage you to look into the paper for SCAT if you're interested in this, but I don't want to get too much into detail on those here. And when it comes to annotations, I mean, the thing is that for these hand-designed filtering and weighting functions, you can usually really only take into account a very limited number of annotations. And often what people are doing is using this tool called VEP or something similar to it, which is, so VEP stands for Variant Effect Predictor. And what it does is it uses genome annotations to get to variant annotations. So genome annotations means that someone has annotated, okay, here's a position for, here are different proteins in the genome. And here's the, the transcription start site. Here's like a regulatory region for that gene. Here's the transcription start site and the untranslated 5' untranslated region. And then there are the exons, the introns, et cetera. And going from all this information, you can get lots of different annotations of what the variant is probably doing at the molecular level in a sort of simple sense. But then there are lots of other tools that people have developed. For example, there are like machine learning tools that tell you about effects on alternative splicing, which if you're not familiar with what that is, there are coding regions of a gene called exons. There are non-coding regions called introns. And, you can have different, before the protein is translated, these exons are spliced together. But you can have things like maybe an exon gets skipped, or maybe the intron is retained, or you can have different kind of variations on what happens in the splicing. There are also tools like Primate AI and Alpha Missense, which will tell you things about the protein structure. And for example, if you have a variant that changes an amino acid in your protein, then you can get some information about how much this change in the single amino acid is going to affect the structure of the protein. And then we use a bunch of other tools, which I can't get into all of them right now, but they do some related things. Well, I'm just listing these as the additional tools we use in DeepRVAT. And our motivation for developing this model is to get past these limitations of standard methods that I mentioned before. So, you really only use these annotations within these manually defined schemes for filtering and weighting. And we want to take sort of all the many different annotations that you can get and learn how to integrate them all together in a data-driven manner. The other thing about these models is they're completely linear, so they assume additive effects. So, if you have 2 variants, it's always twice as bad as having 1 variant. unless you hand design a scheme for taking that into account. But we also want to sort of understand nonlinear and epistatic effects. So nonlinear, but with nonlinear effects, I mean, maybe there are nonlinear relationships between the different annotations. With epistatic effects, I mean, maybe there are, interaction effects between the variants themselves, as opposed to just additive effects. And these models, in particular the variance component tests that I told you about, which are much more powerful for association testing, but they basically have limited use for phenotype prediction. And we'll see later that some of the linear things don't work so well when you try to do phenotype prediction with rare variants. And we want to improve that with DeepRVAT. Okay. So I'm going to get into the model now, and I'll give you an overview and try to explain, also how each of these components works, and then how we train it. And crucially, how can we apply deep learning and not lose the statistical rigor we get from linear methods? So this is the general overview of DeepRVAT, which is, as I mentioned, based on deep sets, which were published in machine learning terms a lifetime ago at NeurIPS. But the way the model works is we have the core component is this thing we call a gene impairment module. And we use this to predict single score per gene, given the variance that an individual has in that gene. And we feed this through a second module, which predicts phenotype. Yeah, we do this in sort of a multitask setup. This is the gene impairment module itself. Let me actually get a little more into each of these bits in turn. So this is what it looks like if you feed in if you look at the level of a single individual. So the first thing we do is we select a set of what we call seed genes, and these seed genes are specific to the phenotype, and they're selected using conventional RVAT methods. And the reason we do this is because if we tried training this thing on all 20,000, roughly 20,000 protein coding genes in the human genome, but this is very, very, very noisy. So, per phenotype that we look at, there may be something between 2 and 50 genes that are actually relevant to it. And so trying to get the model to learn which genes those are from 20,000 genes is pretty tough. So we pre-select these using just kind of linear methods, and then train the model on genes where we expect there to be less noise that it has to sort through. So we select these seed genes. They're different for each phenotype. And also in each seed gene, every person is going to have a different number of variants and annotations. A different number of variants in each gene. And that's one of the, And so when we're training this thing, we feed all of the variants for each gene into this gene impairment module. As I said, we get out a single score per gene. We combine these scores into a phenotype module, and there's a separate phenotype module for each one. And that gives us a prediction for different phenotypes. And as I said, we train this sort of simultaneously to predict multiple phenotypes. And the trick here is that the weights of this gene impairment module are shared across all the different phenotypes and across all the different genes that we look at. So we're trying to learn a function that gives us sort of a universal scoring function that will work for any other phenotype and any other gene. Okay. And the way this module works in detail is, as I mentioned, it's a deep set network architecture, which we chose because it has essentially two advantages for us. One is you can feed in an arbitrary number of variants. And the second is it's invariant to the order in which you feed in the variants. So the first is necessary because we don't know how many variants someone has in a given gene. And the second is useful just because we're not really taking sequence information into account in a direct way. That would be much more computationally expensive and also would require a more complicated model architecture. And given that the data that we have is, it's on hundreds of thousands of individuals, but it's very sparse. Most people don't have a rare variant in any given gene. So we have actually used a relatively simple architecture with just about 17,000 parameters. So the way this works is, we feed annotated variants through this network that we call phi. We also tie the weights together of phi for different variants. We boil this down to a lower dimensional embedding, and then we use depending on the implementation, we use a sum or a max, which is essentially a symmetric or a permutation invariant function here. And to give us an embedding for the gene, which we feed through a second MLP network called rho, and that gives us a single score. So this is now gets us down to a scalar per gene. Okay. And the way to maybe conceptually think about this is that we are bridging between molecular and phenotypic effects. So if we think about it like, we have someone with, here I'm depicting just 3 genes in a schematic way, and somebody has different variants in those genes. We annotate those variants with different effects. Maybe we have missense variants, maybe we have frame-shift variants or peel-off variants, splicing variants. And then this is essentially, so these are models that have been trained on other data. They have nothing to do with DeepRVAT explicitly, at least not in their training phase. But they're telling us something about the molecular effects. And when we try to get to some person's phenotype, we can use DeepRVAT to essentially take in these variants and say, okay, this is, this gene is moderately impacted. This gene is not very impacted. This gene is highly impacted. And then finally, to understand, how strong is the association with the different phenotypes that we're looking at. I heard a bit of a beep.
Right.
Does that mean that there's a question in the...
Yes, that's right. Can we please take it a slide back?
Yes, of course.
Yeah. How do you decide the variant embedding dimension?
Well, I want to say we did a really sophisticated hyperparameter search. Actually, we tried out a small network at the beginning, and we never were able to do any better. So we sort of said, okay, we started out with something linear. Results look promising. We said, okay, let's make it a little nonlinear. Let's grow the network a little bit at a time. And I had so that my collaborator on this, Eva, who I'll mention later, was a master's student at the time in the group. And she made a sort of, guessed at an architecture, and it worked well. And then we did a bunch of sophisticated hyperparameter searching and changing up the architecture and things like that. And we were never able to do any better. So we just stuck with what worked.
I see. So for example, what would you expect to happen?
Let's say, sorry.
I see. Sorry. What would you expect to happen if you used, for example, instead of any sort of encoding to obtain an embedding, you would just go directly for a dimensionality reduction method and just do an additive operation over the annotated variants? Would it behave in
That's
a similar
good question.
manner?
So you mean do it in a fully unsupervised way?
That's right. That's exactly right.
It's a good question. We haven't tried it. I can't tell you what would happen. It might be, we did think a little bit about maybe, like a self-supervised thing. As I mentioned, we don't look at a lot of different variants. So maybe like combining what you mentioned for those variants that we don't look at together with this supervised learning approach might be interesting. We have done some thinking about that, but no results that I can tell you.
Okay. Thank you for the input. We have another question from the audience.
Actually, this a follow-up question from Suzal's question about the individual modules for the annotated variants. So you did mention that you've implemented weight sharing, right?
Yeah.
So is that also something that you looked into, either untying the weights and have them be variant specific? Maybe that'd be a bit wasteful, or is it more like, I think you mentioned like the more deeper interest in the weight sharing, where it's more of a universal representation of different variants for a given gene.
Yeah.
If the latter, could you expand on that a bit? Because I guess if I could add a note to that, there's like for instance, in a previous reading group presentation, there was this idea that you could use orthologs across species to kind of generate this augmented representation, right? Almost as if you have orthologs from different species, it's different views of the same gene or RNA transcript. So I feel I'm tempted to think of this in the same way, where for a given gene, these different variants are almost like different views, but of the same gene, but of course different clinical and phenotypical downstream implications. So I was just wondering, and it's kind of an open-ended question, I know, but just more on your thoughts on that.
Yeah, for sure. Maybe to the second, well, part of your question I think will be answered in 2 slides, or well, 1 slide from where I was before. So I encourage you to ask it again if it's not clear after that, in terms of why do we want it to be universal across different genes. So I think we might be able to, I might be able to answer that, which is not, and but maybe one point. So I think making it variant specific is not really possible in the setup that we have right now, because it doesn't actually know anything about the variants except for their annotation vectors. So we sort of annotate the variants as a pre-processing step, and then they're just represented for the model as an annotation vector. So if 2 variants have the same annotation, it has no idea which one is which, right? But what one could think about, and we actually have a grant application for this, and if anyone you know is looking for a PhD project and is interested in thinking about this sort of thing, they should reach out to me because we're hiring in the fall. What we are thinking about is how to take what we've done, and I understand I haven't explained that yet, but take what we've done and make the sort of the scoring, or at least the the embeddings for the gene phenotype specific. Because as you said, there may be a case where, let's say I have phenotype A and it's sensitive to splicing variations in this gene that I'm interested in, and I have phenotype B, and this is actually sensitive to loss of function variants in the gene that I'm interested in. And we don't capture that yet, but we would like to in the future. Again, we are hiring. So I hope that answers your questions. I'll go ahead 2 slides and explain, and then I'll check in with you if the next slide answers your question.
That's very interesting, but I will probably follow up in 2 slides. Thank you.
And just a little tack on to the discussion. If you need us to facilitate the links to your grants or your groups, Brian, please do let us know, and we'll happily share them across our social media and such.
Ah, cool. Thanks. Yeah, I will. I'll post in Slack once the job ad goes live.
Wonderful.
Cool. Appreciate it. Okay. So here is how we now do. So I talked about training. So how do we do association testing, and how are we going to maintain statistical rigor without losing power from splitting up our data? I mean, one thing we could do is just cut the data in half, use half of it for training and half of it for association testing, or maybe 70-30 or whatever. But we didn't want to do that. It turns out association testing is very sensitive to your sample size, so you can lose a lot of power by doing that. We tried it, doesn't work very well. So we came up with this scheme instead, which is essentially a cross-validation scheme with a little bit of, if you squint at it hard enough. So as I mentioned, we train on these sort of pre-selected genes. And what we do in this cross-validation scheme is we split our data 5 ways. And we train on, the first 4 full cross-validation folds and these C genes. We do nothing with the other genes. These are just ignored for now. And then on the held out samples, we compute the gene impairment scores. And this is why we want to have a universal, this is one of the reasons we want to have a universal scoring function, because we never actually look at these genes during training. So we have nothing, we know nothing about them because they're not related to any of the phenotypes that we train on. And they might not be related to any known, they might not have a known relationship to any phenotype. So basically, if we want to be able to score any gene, we need to have sort of a universal function. So we compute these gene impairment scores here. And then we start filling in a matrix of pre-computed burdens, right? And then we just do the standard CV thing, which is we walk our test set through the data set until we've filled this matrix entirely. And now what we can do is, columns of this matrix are genes and rows are samples. And to do rare variant association testing, all we have to do is extract a single column of the matrix, which corresponds to the gene, compare it to the phenotype, see if we get an association, and that's it. And in fact, for UK Biobank, we trained this on some set of phenotypes, and then we looked at some other phenotypes, then we wanted to look at some more phenotypes. But in essence, we really just did the training once. Okay. ignoring our development cycle, but we did the training and scoring of genes once, and then we could look at whatever phenotypes we wanted. So basically, I mentioned that the gene scores are gene agnostic, right, which is important to fill in these genes that we never saw during training. And I mentioned that they're phenotype agnostic, which is important if we want to look at, like, for other people who are looking at who are using DeepRVAT, they don't have to go and retrain the model. They can use it to look at any phenotype that they want, and I'll give some evidence that this works in the next slides. I hope that answers your question now.
Yeah, it kind of does, because of the, so if I understood clearly, you kind of have these, sorry, just to confirm, is the training set related to the seed genes that you were talking about with their variants, or is that, am I conflating two things?
Exactly. So the seed genes are is this subset of genes that we use for training. And then we also use a subset of samples. So we're subsetting in two directions for the training, in 2 dimensions. Yeah.
And okay, so I guess in saying that this would answer my question is by saying that once you filled up this matrix with sort of let's say imputed impairment scores, they're not imputed, imputed. Then you compare those with the phenotype, but then for a given column, which is a given gene, that gene has one phenotype. But in my mind, like or again, maybe I missed a little step there.
Well, okay, during training, the gene has one phenotype, although you can use the same gene for multiple phenotypes as well. That works. So during training, but then at testing time, you can put any phenotype you want here. So the gene scores stay constant. So I just have a matrix that's samples by genes. So in UK Biobank, it's 500,000 by 200,000. When I want to do association testing, it's just so I have this matrix. You're interested in some phenotype. You give me the phenotype values for all the samples, right? And then I compare them to the gene scores and tell you if there's a statistical association.
Yeah. Okay. So yeah.
And I can tell you gene by gene whether that's the case, right?
Yeah. I guess what I was confused about is a bit silly, because the point is you want to see which genes are associated to phenotype. It's not like there's a phenotype per gene. But actually, my question was, maybe if I could clarify, is more back to this idea that for a given gene, if you have different variants, those different variants could have different phenotypes, right? And that harkens back to the question I had 2 slides ago. So would this framework be adaptable for in the case where you would have you would know that you'd have different phenotype per variant for a given gene, or that you hypothesize that there may be maybe is a better way of putting it?
What we could adapt it to, so I think like adapting it to a different variant per gene might be hard unless we extend it to common variants where you can really look variant by variant at what is happening. What we could do is adapt it to sort of like an annotation, having annotations having different importance per phenotype, if you see what I mean. So like maybe splicing annotations are more important for one phenotype, but missense annotations are more important for another phenotype. That is part of the follow-up work that I mentioned.
Okay, but in case of, you just said it could work for common variants. I guess for rare variants, then
the problem is for rare variants, like I just don't see them. If I want to know, like, if I really want to know variant to phenotype associations, I just don't see the rare variants often enough to do this without some kind of intermediate representation. And that's what these annotations give us. They give us an intermediate representation that's sort of like, it's still quite rich, but it's lower dimensional than saying than a 26-dimensional one-hot vector or something, right?
Got it.
A 26 million dimensional one-hot vector.
Excellent. Thank you.
Makes sense? Yeah, cool. All right, so let's get to some results. I'll try to pick up speed a little bit. Hopefully it was justified to spend a little bit more time on the method, a little bit less time on the results. You will see our bars are the best bars, but yeah, I'll describe what the results are and why you should care now. But again, trying to pick up speed a little bit. Here's the application to rare variant association testing. So what we do here is we compare this to different models. So in the blue bar, you see this thing, which is combining the burden and scat test that I described some slides ago. This is sort of the classical approach. But these don't use very many annotations. They essentially just use the allele frequency of the genes of the variants to weight them with sort of the hypothesis that variants that are more rare have larger effects. We compared it to two methods that actually do use many different functional annotations of variants, but these are super complex. These are linear models. They're similar to the scat tests that I mentioned before, but they're super complex, like meta models, that they do a bunch of different tests and they use some statistical trickery to combine the tests into a single p-value and so on. They're relatively complex and they're very slow to run. And we compared this to our DeepRVAT burden score, which is really just a single score per gene. So it's very related to these burden scores that we mentioned before. And it's extremely computationally efficient. And we did this on 21 quantitative traits from UK Biobank. These are the same traits that we trained DeepRVAT on. And on these traits, we outperform even these complex models. And what's very encouraging, actually, because, of course, we just, when I say outperform, it means we're looking at the significant gene trait associations that we found. But we don't just want to find more associations. We want to know that we can trust that these associations are real and not just, overfitting, spurious discoveries, so on. So what we did was, here we had used 161,000 individuals. So there was like an earlier release of the whole exome sequencing data set from UK Biobank that had 200,000 people. Then just to keep things simple, we subset to the unrelated individuals with European ancestry, or at least predominantly European ancestry. I'll come back to ancestry in a second, because we're not just interested in discovering things to improve white people's health. What we did was we looked at studies that were on larger UK Biobank cohorts. So eventually UK Biobank released 470,000 whole exome sequences. And so there are these studies that do it on larger cohorts. And these are using essentially the classic methods that I mentioned, but since they are on really large cohorts, they have a lot better power to discover things. So they have more discoveries than any of these methods on the 161,000 individuals. And what we can do is we can look at, okay, how well do the things that we find replicate in these other studies? Which is essentially telling us, if you start with a subset of the data and find something, and then you increase to a larger subset, do you see that your original finding was just kind of noise? Or like a false positive, or does it hold up in the larger subset? Encouragingly, not only do we discover more things, but the things we discover also hold up a little bit better than even the best of these methods. So that's one thing. But I mentioned we wanted to have a score that was actually universal in a sense, right? So this is kind of maybe an indication that it's essentially universal across genes. How about across different phenotypes? Well, what we did is we took 13 additional quantitative traits. By quantitative traits, I mean things that take on a continuous value, like these might be your cholesterol level or your height or your weight or something like that. And we looked at these and we compared it to the other methods, and we still do better when it comes to what we discover. In fact, we even perform a little bit better in comparison on these held out traits than we did on the training traits, which is quite encouraging. The replication looks similar. I didn't put it in the slides, but you can check the paper. And then we also looked at not just quantitative traits, but we looked at disease traits. And here we're only comparing to sort of one of the standard tools, which is called Regini. We actually integrated DeepRVAT with Regini to get these results. And we didn't compare to the others because the others give just tremendously inflated numbers of associations. I'll show this on the next slide. But yeah, what's really nice here is that we have an especially large increase when it comes to these disease traits, which are anyway what you're what you're really interested in. The quantitative traits are interesting too, but the disease traits are arguably more interesting. And here we get a really, really big increase in the number of gene trait associations that we can discover compared to the standard methods. So what is this tool that I mentioned called Regini that we integrate with? It's a relatively new tool for doing classical statistical analyses in genetics. What's great is that we can really seamlessly integrate with it. It's just kind of take whatever we output and put it into the right data format. And then we feed this into Regini. It gives us a lot of things for free, like it can handle imbalanced case control ratios. So if you have a pretty rare disease, so you have only like a small number of individuals with the disease in your cohort, Regini can handle that without giving you a bunch of false discoveries. It can handle population structure, for example, if people are related, or even multi-ancestry data sets. And it's very fast. So we can get our association testing results on these 34 or 63 phenotypes, whatever we tested on, within about an hour. You can see that there is actually there's definitely a big benefit when you increase the sample size going from 200K to 500K. And there's actually quite a benefit when you go from the 500K European data set to looking at all ancestries. So there's a lot of, these are just the number of discoveries that we make on these different data sets. And it's quite important to take all of this data into account simultaneously. And as I mentioned, if you're familiar with QQ plots, you'll see that these methods, the methods called Monty and Star on the previous slides that use many annotations, they lose their calibration, meaning they make a bunch of false positive discoveries when you go to these disease traits, especially when you have imbalanced case control settings. Perhaps if you're not familiar with QQ plots, you can look here and you can see that weirdly, as the number of cases of the disease you have gets lower, the number of discoveries that these tools makes gets much higher. So this is on a log scale. So these tools are telling you, 500 or 1,000 genes are associated with a very rare disease. Usually it's, you would imagine like 1 to 10, something like that. But DeepRVAT stays quite reasonable in these settings. Okay. We tried to look into some of these associations in a little bit more detail to see if we could find something new, even though UK Biobank, well, it's been quite picked over as a data set. People have studied lots of things on it, but there were still a couple of new biological insights that we could find. And I'm just showing here our favorite one. So we found this significant association between heart failure and ASXL1. It has a Bonferroni corrected p-value of 0.01 roughly. So not super significant, but maybe showing that, there's but it's robust, I should say. And then it's maybe showing that there's a kind of, there's a value to this increased sensitivity that DeepRVAT gives you. So these things, this would not have passed a significant threshold with other tests, but does with ours. And it's actually, this gene has been previously implicated in heart failure because it's a driver gene of something called clonal hematopoiesis, which is a precursor of blood cancers. It essentially means that your bone marrow stem cells start to accumulate mutations, which means that your blood is filled with a bunch of different clones. That is, it's cells with different genetic makeups. And this kind of, I'm not a biologist, as I understand this wreaks havoc in your body, and it can have a negative effect on your heart. And people have found experimental, both genetic and experimental evidence that somatic variants in ASXL1 can be related to heart failure via this mechanism. But what we're showing here is that we believe we have the first evidence for effects of germline variants in ASXL1 on heart failure. And just to test that we're not sort of just more sensitively picking up on effects that are, sorry, I'm going to close the window. Church bell is going off. So just to check that we're not just sort of more sensitively picking up on known effects. We looked at all of the people with relatively high DeepRVAT scores in our data set and checked them for whether the variants that were driving the DeepRVAT score are known to be implicated in clonal hematopoiesis or related diseases. And for the top ones, which are loss of function variants, mostly loss of function variants in those genes, it turns out that they are, but there are also quite a few variants that are new. And these, we think, are candidate variants that could also be implicated in clonal hematopoiesis and various blood cancers. Okay, just as a last result to show here, we also used DeepRVAT to do phenotype prediction. So essentially what we did was we looked at a common type of score called a polygenic risk score, or PRS. And so this takes into account common variants, like I mentioned, which are studied statistically. And then we added DeepRVAT scores for phenotype relevant genes on top of that. And we're looking at the same quantitative traits that we did in the previous slides, and we saw that you can actually improve the variance explained for those traits by genetics by including these DeepRVAT rare variant scores. You'll notice the y-axis here is quite modest in scale, so we get a really a somewhat small increase in performance when you're trying to just predict the phenotype. But where rare variants seem to have a bigger impact is if I want to predict the extreme phenotypes, so I look at the people in the bottom 1% or the top 1% of the distribution. So for example, if you're in the bottom 1% for good cholesterol or in the top 1% for bad cholesterol, you might want to know this because you may have an increased genetic risk for heart disease. DeepRVAT is doing much better than other scores here, and actually, adds something significant to the prediction. Yeah, it's at least for this bottom 1%, it's the only one that's sort of consistently above 0% improvement on scores. Yeah, I think we have a question.
If you had to attribute that increase in performance to any one component of the model, what would you put your money on?
Well, we did two things here. So one is we checked, all of these scores are part of DeepRVAT. So there's something to be said about combining them together. I think that's the main thing that I would say. You get a little tiny boost as well because in this DeepRVAT score, we additionally added in like the knowledge that DeepRVAT generates about phenotype relevant genes. That gives you a little boost, but the main boost is in combining these different annotations together into a data-driven score.
That was my intuition as well. Thank you.
Sure. Okay, so very quickly through the summary and outlook, as I see I'm running out of time, but yes, so this is the first method that uses deep neural networks to learn nonlinear effects from annotated rare variants in rare variant association studies. As you've seen, we came up with methods to retain statistical rigor, even though we're using deep learning. And we've really increased the power to discover gene trait associations. It's more reproducible than other methods. And we can apply it to multiple downstream tasks, which included phenotype prediction. And we also saw that it generalizes to new phenotypes. So what are we working on now? Well, some of the things we're working on are applications to new data sets. So for example, we're interested in pediatric cancer, where we expect germline. We're part of the German Cancer Research Center. And we have some data sets on pediatric cancer where we expect germline variants to have a larger impact, although we are also interested in applying DeepRVAT to somatic variant data. We also think it could have a big impact on rare disease. And one of the big things we're working on right now is working on a method extension so that we can do, because right now we require individual level sequencing data, which is quite hard to get a hold of on a large scale, and also to combine all this data in one compute infrastructure due to data privacy. And so we're working on extending DeepRVAT so that it can be used in a sort of meta-analysis setup where we use summary statistics that we can learn from UK Biobank and combine this with smaller targeted disease cohorts to study these rare diseases, which are not so easily found in UK Biobank. And we're also interested in looking at data sets like All of Us, which at some point will be larger, or should be larger than UK Biobank, and is also ancestrally more diverse, which is quite interesting to us. Yeah, we're also looking at taking DeepRVAT-like ideas, maybe not exactly DeepRVAT, but similar ideas, and applying it to the non-coding genome. And we have a very interesting data set that's being collected right now, which is called Cardinal. It's from the UK Biobank and East London Genes and Health, and we're part of this consortium. We have about 5,000 donors from UK Biobank with single-cell RNA sequencing data, and we have the link to everything that you can get in UK Biobank. We also have 1,500 donors from another study called Genes and Health, which, in contrast to UK Biobank, which is about 80% European ancestry, these are all people of South Asian ancestry. Yeah, it also has links to interesting additional omics layers and phenotypes. As I mentioned, we're hiring, so if you're interested in sort of expanding DeepRVAT to larger sets of annotations, incorporating it with other sort of foundation models, language model, biological sequence language models, that sort of thing, I encourage you to reach out. And as I said, a job ad will be online in the next couple of weeks. Yeah, and finally, I want to acknowledge the great groups at DKFC and TOOM that we collaborated with, and please check out the paper and our GitHub repo if you're interested. Thanks very much.
I would like to take a moment to thank our speaker for the wonderful presentation. And with that, open up the floor for any additional questions.
I have another question. So, I mean, you opened up at the end with the non-coding genome. So you work really predominantly with whole exome data. So purely sort of coding variants, is that a fair statement? Or something that
Almost. There is some overhang. So, there's a little bit of overhang when you do in any exome sequencing kit. And also the kits don't exactly line up to the genome annotations. So there's a little bit of there are some non-exonic variants, and some of the annotations do capture interesting things about them. But yeah, it's largely coding variants. Yeah.
You motivated like many of the rare variants being like in the non-coding genome, and I can imagine sort of the variant annotations being a lot more challenging for non-coding mutations. Yes. So just do you have how do you envision sort of integrating whole genome sequencing within this framework?
Yes, good question. So yeah, it is definitely true that sorry, I'm not sure if you maybe misspoke, but actually non-coding variants tend to be more common. Coding variants tend to be more rare. I think you said it the other way around, but I assume it was just me speaking. So that's one thing. But so like when we look at rare variants, there's some justification for looking at the coding region. But yes, you're absolutely correct. I mean, in the non-coding region, it's harder to do annotations because it has to do with relatively complex regulatory logic, which we don't understand as well as we understand effects on coding regions. So you're completely correct about that there are additional challenges there. So what we're trying to do is take approaches that with the annotations that we have. But I agree. Like one of the interesting things will be, can we improve the annotations that we have in non-coding regions, and that will make tools like DeepRVAT or anything similar, much more effective. So yeah, you hit on a good point. But I don't have an answer for how to solve that. It's just I think it's a big research program to do so.
Thanks.
Do we have any more questions?
Yeah, we want to hear from David.
Hey, thanks. Super interesting stuff. I had a question. Maybe it's something you get sometimes. Have you considered, or do you have any plans to see if you might have if your tool performs equally well on different populations? Because you did a lot of stuff with the UK Biobank but that's primarily European individuals, and so we know mutations might have different annotations in different populations, and so is this something that you've thought about? Mm-hmm.
Yeah, that's very true. I mean, largely we expect rare variants to have less ancestry-specific effects, and that is actually borne out in not just in our study but in others. I mean, you did see that like things actually go quite well when you jump to looking at all ancestries. But yeah, I think we would need other data sets, like I mentioned, in order to study this in more detail, just because UK Biobank is not super diverse. And I would just say generally that with common variants, the problems are larger because often it's in sort of, as I mentioned, sort of subtle regulatory logic. So it might be that you have a variant in a transcription factor binding site. But then there are compensatory mechanisms. For example, that transcription factor is upregulated by some other variant in the genome that's co-inherited, or you co-inherit a different variant at another transcription factor binding site that balances out the effects, right? So common variants have sort of relatively complex architecture when it comes to ancestry effects. Rare variants are a bit less. You expect to be a bit less ancestry dependent, because if you lose function in a gene, so for example, if you have like a stop mutation gained 10% through the gene, then you don't expect to have a functional protein, and this should have a somewhat similar effect, no matter what your ancestry is. But you do still see some effects, like in these Bangladeshi and Pakistani ancestries, for example, some people have even homozygous loss of function variants. And it's even relatively common in the population. But then there are, compensatory mechanisms elsewhere in the genome that keep them healthy, even though they have an entire gene that's not functioning. So there are some ancestry effects, but they're just less prevalent than common variants.
Do you think so? This I don't actually know the answer to this. Would it be possible for a variant to be low frequency in one population and but maybe higher frequency in another population, and then that could affect maybe the results? Mm-hmm.
This definitely happens, and it's a challenging problem. Just a lot of studies, they will just do their association testing in an ancestry by ancestry basis, and they don't even try to combine them because of exactly these problems that you mentioned. That the allele frequency of a variant, which is often used as a proxy for how impactful it's going to be. It can vary between populations, and a variant that's rare in a European population may be common in the African population, but that doesn't mean it's harmful in the African population. It just means someone had a mutation tens of thousands of years ago, and it spread throughout the population. But it's fine, right? I mean, it doesn't mean it's harmful. It just means that it hasn't spread in the European population. So yeah, it's an excellent point that the allele frequency is a tricky thing when you go into multi-ancestry studies. And it gets even trickier because not everyone is purely, most people are not purely European or purely African or purely East Asian. There are many admixed individuals.
Okay, cool. Thanks. And thanks for the talk again. That was really great.
Thank you.
Thanks, David, for that question. You're good. I had to step out for 5-10 minutes to get some food for the people here. So I might ask a question that you might have already answered.
Go for it.
So a lot of, if you do GWAS studies of many diseases, the traits are very, not the traits, but the signatures are very polygenic, right?
Yes.
So most complex diseases have a bunch of different hits, and you might combine them into polygenic risk scores. Mm-hmm. Could you leverage something similar here, right? Where you would have different gene impairment scores that you would combine to then associate to a phenotype? Or yeah, okay, I did. I guess I did.
Yeah. This is when you stepped out. So I can explain this slide in a little more detail anyway. So we did actually take polygenic risk scores like you were describing, and we used these to predict these different quantitative phenotypes that we were looking at. And then what we did was we looked at the genes that we found to be associated with those phenotypes. And we added on the gene impairment scores, so the scores coming from rare variants. So we added for each gene, we added one predictor to our, and then we just built a linear model to try to predict. So the PRS is one predictor plus, age and sex, and then genetic sex, and then these rare variant rare variant impairment scores. Yeah, so you do get like some additional benefit from that. As I mentioned, it's when you're trying to predict for the entire population, it's very modest, the benefit that you get, because most people don't have a rare variant in the relevant genes. And even if they do, it's often just kind of not a very impactful one, just kind of a random mutation that doesn't do much. When you look at the extremes of the phenotypic distribution, so bottom 1%, I'll try to remember which is the good cholesterol and the bad cholesterol. So I think bottom 1% of HDL cholesterol is bad for you. Top 1% of LDL cholesterol is quite bad for you, right? So when you try to predict your genetic risk for being in one of these categories, the rare variants have a much more impact on the quality of your prediction. Which you would expect, because if it was a common variant that shot your cholesterol through the stratosphere, it wouldn't be very good for humanity. Right.
You suspect that for certain traits that combining common and rare variants, but the rare variants haven't been sort of embedded through RVAT, would that potentially help even the ones that are successfully modeled by common variants, then adding in the rare variants? Maybe do you think that could help sometimes? Or?
Yeah, I don't think we tried that for polygenic risk scoring. It's a good question. We tried it for association testing, and you get massive inflation in the number of discoveries that you get. We tried to investigate a little bit. So like essentially just take DeepRVAT, but instead of feeding in rare variants, just feed in all the variants and then try to do gene trait associations. We tried that. You get ridiculous inflation. I mean, it tells you there are 3,000 genes associated to cholesterol or something. It just doesn't make any sense. We did a little bit of poking around before we gave up on it, but our hypothesis is that it has something to do with linkage disequilibrium, which is essentially correlation between variants, which is small and more or less negligible when it comes to rare variants. But when you get into common variants, you really have to be careful about linkage disequilibrium. And we didn't come up with a great way of dealing with it in DeepRVAT, and sort of, for the moment, dropped the idea. But we have it on our sort of backlog, so to speak, to come back and take a different modeling approach with it.
Okay, so a lot of false positives if you combine those.
If you naively just feed all common variants into DeepRVAT, you get crazy numbers of false positives. Not a good idea. I can save you some time if you were thinking of trying that.
All right. Well, we've taken we're 11 minutes over, so you've been generous with your time. Thanks a lot. And also, by the way, you extended your apologies, but us too, by the way. We completely forgot that that's a common issue here in Eastern Canada, where we change our hours a month ahead of time of other people. So that was on us, definitely. So
yeah, no worries. I mean, you can probably hear I'm from the US. I should have known, but it's just, so I've been living in Europe a long time, and it slipped my mind.
That's understandable.
So, yeah,
for sure. So thanks for this take two. Very much appreciated. So,
yeah, I appreciate the second chance.
Wonderful. Thank you very much for the talk, Brian. And that concludes the 16th Multiomics Reading Group. This stops the recording. Thank you very much, everybody, for attending. And Brian, once again, for the wonderful talk.